I'd now like to invite our final speaker for this session, who's going to move away from pneumococcus and into something entirely different. Well, not entirely, but in a way.
Thank you, Bill, and thank the organizers for inviting me for this excellent meeting. And this talk has almost the same title as your talk, Bill, and in the end, you will hear that I have almost the same message as he had. So this is the view from a clinician confronted with antibiotic resistance, and from my talk you will hear the acknowledgements. That's Rob Willems, the molecular biologist in our lab, and Martin Bootsma, mathematician, and I'm actually more or less speaking about their work. I will talk about two pathogens, the gram-positive ones. This is Enterococcus and Staph aureus. I would have loved to talk about the gram-negative bacteria with the ESBLs and the KPCs, but to the best of my knowledge, there's hardly anything known about these bacteria now what relates to the title of this presentation. So why Enterococci? Well, this is the reason why. This is in time what happened in our hospital with the bloodstream infections, the number per 100,000 patient days. And as we can see in time, some were increasing, but the one that was most rapidly increasing is Enterococcus. What are Enterococci? Enterococci are opportunistic nosocomial pathogens. They live not only in us, but mostly everywhere where you can look at them. They are intrinsically resistant to many antibiotics, but not to ampicillin and vancomycin. And these were the, let's say, the most important ones. But then in the late 1980s, in the previous century, things started to change. And in retrospect, it was clear that there was an emergence of beta-lactam resistance among Ephesium clinical isolates, which actually more or less happened first in the United States. Nobody took attention of that, but then in the 1990s, it was clear that there was a change in the Ephicalis to Ephesium ratio. So these are the two most important species within the Enterococci. And Ephicalis always was the most important one, but then Ephesium started to emerge and things started to take notice, or the Enterococci got notice when there was a rapid emergence of vancomycin-resistant Enterococci, especially in the United States. And vancomycin was the, let's say, the last antibiotic to treat them with, so that's when people got worried. These are two graphs which look more or less similar. This is data from the United States from 1981, and this is the proportion of Enterococci from bloodstream infections in ICU patients. This is the proportion that was vancomycin resistant, was absent before 1990, and then rapidly emerged to these levels. These are data from the Netherlands, from our hospital, same graph, so it's a proportion of invasive infections in time. This is not vancomycin resistance, it's just ampicillin resistant Enterococcus faecium. The bacteria are very much alike, with the difference that these are still vancomycin susceptible, but we saw a similar increase, although 10 years later. So Rob Willems did a lot of work on it, and he collected many of these VRE strains from all over the world, and we did multi-locus sequence typing, which has been explained by Bill. And then we looked at clonal complexes, and we found distinct clonal complexes related to the host where the Enterococci were harvested from. For example, the animal strains, because VRE was very frequently found in animals in Europe, related to the alpha parsinus. Animal strains clearly were distinctly located than this clonal complex 17, because all the strains that we got from hospital outbreaks in 5 continents, they're clustered together here. This was multi-locus sequence typing, e-burst analysis. Everything looked very simple, and Rob Willems continued to, let's say, collect all these strains, and this is a e-burst analysis of 554 different sequence types, and this is 10 years later. And now the question is, can we still speak about clonal complex 17, because it starts to accumulate more or less everything. There's still distinct groups that can be identified, such as these, that they all come from veal calves. This comes from poultry. This is from pigs and humans. And this is from humans, and then it's vancomycin susceptible Enterococci. But then there was this paper which more or less confirmed what Rob was already thinking, that there is a lot of inaccurate clustering here, and can we really speak about clonal complex 17 anymore, because the e-burst probably is not the best typing scheme or analysis method if there is a lot of recombination of genetic material ongoing, which we already knew was the case in Enterococci. So the analysis then of the answer to that is to do a BAPS analysis. And there are people here in the audience that know much more about that than I do. But that was done with these data. That paper is now in preparation. And based on the BAPS analysis, it was that there were 7 clusters and populations that could be subdivided in actually 14 subgroups. And these are the 7 subgroups, and I will tell you a little bit more how this now relates to what we initially thought of the clonal complex 17 as, let's say, the most successful clone in hospital settings. Well, actually what we now think that clonal complex 17 is more a combination of 3 sequence types, 17, which was still the founding father of the clonal complex 17, but also 18 and 78. And these 3 are still related to, for example, the hospitalized patients. So they are more frequent. This is the odds ratio of these analyses. They are more frequently found in the hospitalized patients and less frequently found in farm animals and meat. But there are some groups in between in which maybe there's interaction between these reservoirs. So still we think that there is a distinct group of Enterococci, Enterococcus faecium, which is very successful in hospitalized patients, causing outbreaks, which is clearly distinct genetically from the enormous amount of Enterococci that we find in the animal reservoir. What is also obvious from the data is that there is a shift in time. Lineage 17, 18, and 78, and all the data that Rob Willems has is that we can clearly see that this lineage 78 is rapidly emerging now in the latest years and was more or less absent in the beginning. So there seems to be some time effect ongoing. And these data more or less confirm previous analysis from Helen Levis. She did a micro-based phylogeny of all these Enterococci that we had and found this group which contained all the CC17 isolates, but now appears to contain all the BAPS analysis of these 3 groups. This, more or less in retrospect, corroborates very well with the new BAPS analysis. So what then does explain the success of this, let's say, clonal complex in hospital settings? The answer simply is we don't know. 5 years ago, hardly anything was done with Enterococcus faecium when it comes to molecular biology or with virulence traits, whatever. In the meantime, many potential virulence factors have been identified, such as the S gene, SGRA, ECBH, pathogenicity islands, all things like that. And we can see here, if we distinguish the 2 groups as the hospital clade and the non-hospital clade, then we can see here that some of these potential virulence factors are clearly enriched in this population, but up till now we have not been able to really pinpoint which of these factors or the combinations of factors is really responsible for the true success of this clade that we see. And it certainly is not just the antibiotic resistance. So the conclusions from the enterococcal work is that not all ephesium are identical, that non-hospital and hospital-derived ephesiums are genetically distinct when it comes to the core genome, but they also differ in the putative virulence gene content, which is part of the accessory genome. The hospital sequence types represent successful hospital-adapted clones capable of colonizing and invading hospitalized patients and thereby replacing the indigenous flora. That's work that we did together with Fernando Baquero. And 3 hospital-derived clones have a high propensity of inter- and intra-hospital spread, and clones with identical sequence types are found in different countries and on different continents. So it really looks like we are facing a global, let's say, epidemic, hospital epidemic. And it also looks like that this clade has been adapting many different genes, the cumulative acquisition of what we think now more than 150 genes, which looks like a clear case of genetic capitalism. The ones that are strong become stronger, and this is exactly what seems to be occurring here with the ephesium. So if we then move from ephesium to Staphylococcus aureus, methicillin-resistant Staph aureus, the epidemiology has been changed dramatically in the last 15 years. In the United States, with the emergence of community-acquired methicillin-resistant, most prominently USA 300, and in our country, with the occurrence of a clone, which was very frequently found in animal reservoirs, like pigs, but also other animals. This is sequence type 398. And the question is, are these really successful, and can we quantify their success in order to, let's say, to distinguish any success from really, let's say, natural fluctuations in occurrence? So there's global distribution of these, let's say, community-acquired and also non-community-acquired MRSA clones, and I will focus on the ones, the community-associated MRSA first. It's highly clonal in the United States, with probably USA 300 being most prevalent nowadays. There's successful transmission in community with documented spread in sport teams and gay communities and daycare centers. The transmission capacity in healthcare settings is largely unknown, and the route of patient-to-patient transfer there and also in the community is largely unknown. And in contrast to what we did know about MRSA, which was the hospital-associated MRSA, which mostly infected debilitated and older patients, this is mostly affecting and infecting young and healthy people. So these are data from Cook County Hospital in Chicago, from Bob Weinstein, and they had a system of identifying microbiologically documented infections with community-associated MRSA. Many of them were typed, and almost all were USA 300, and hospital-associated MRSA, which were hardly any were USA 300. So this is in time from January 2000 till 2005, and this is a logarithmic scale for the community-associated MRSA, which, let's say, exponentially increased, and this is just a numerical scale for the hospital-associated MRSA. Here they went into a new building, which was rapidly polluted, as you can see. So this is the data that we use, and then we asked Martin Bootsma to try to make sense of these data in using a previously developed model that we used to study or to investigate the different aspects of our search and destroy policy in Dutch hospitals. And this is a multiple hospital model with ward structure and patient-to-patient transmission in the hospital, mainly via healthcare workers. And we added transmission in the community for this analysis, which was excluded here. And he came up with the following formula that based on the epidemic growth curve of community-associated MRSA, he estimated that the R0 value should be 1 plus, and then that's 1.34 times D, and D being the average duration of asymptomatic carriage of community-associated MRSA, which is the critical value here, and which is exactly what Christoph nicely showed for pneumococcus. You should at least have that information, and we didn't have. But if we look at the relationship until the ultimate prevalence in the population at risk, depending on the duration of carriage, we can see that it can become very high. The red line is in the absence of any interventions in hospitals, and the green line would be, well, if you could stop transmitting and actually clear carriage once patients come in hospital. And based on the data it suggests that there's really community transmission, so this is not at all very surprising. We tried to say something more about this R0 value, but actually we didn't succeed in that because there were too many unknowns. We didn't know the duration of carriage, the asymptomatic carriage, or the carriage after treatment. We don't know whether there's competition with other MRSA genotypes in carriage, whether there's competition with susceptible Staph aureus genotypes, whether it's immunity after infection or carriage. We simply don't know, and this was 5 years ago. And to the best of my knowledge, we still don't have the answers to these important questions, especially also not for host specificity, because for the susceptible Staph aureus, we do know that about 25% of the population is never carrying Staph aureus, and another 25% is always carrying Staph aureus. So this information is, I think, critical to understand the epidemiology of community-associated MRSA, and we still don't have these data. So then, our own problem is ST398. This is the strain that we find in the animal. It's highly clonal in all of Europe. It's found in all countries. It has very successful transmission among animals, and from the animals it goes very successful to the animal caretakers. We now know that it from there doesn't go much further. And transmission capacity, that was our big worry in the healthcare settings, is largely unknown. In the Netherlands, we have been very successful in keeping hospital-associated MRSA and other MRSA strains out of our hospital settings with a nationwide search and control strategy in which we screen high-risk patients when they are admitted, and if they have MRSA, they are treated in isolation to make sure that they cannot transmit their bugs to other patients. So Martin Bootsma and Peter Trapman came up with a model to use the data that we have to estimate the, let's say, basic reproductive number per hospital admission for these strains, and then compare that to the other strains that we see in the Netherlands. The setting is that we have very infrequent introduction of patients with MRSA in the hospital, so we can assume that actually the whole population is at risk for acquisition of MRSA. Once a patient is detected with MRSA and was not in isolation because we did not know that the patient had MRSA, then contact screening was performed to determine all secondary cases that were still in the hospital, and there were measures taken to control the outbreak. The input is further that we need the number we get the number of secondary cases, do genotyping on them, so we can define the outbreak sizes. We know the detection rate in the hospital, so if there is a patient with MRSA unknown, we can estimate how likely we are to find him, and we know the discharge rate of patients. And with that information, we published two clinical papers on what we did, and we calculated the RA values of the ST398 and compared that to all the other genotypes that we found, which were all what we called hospital-associated MRSA. There was no USA 300 in that, and found that these hospital-associated strains were 5 times more likely, had a 5 times higher RA value than for the PIC-associated MRSA. And from that, we concluded that probably in our hospital system, this strain is very unlikely to become endemic because the RA value is really very low and much lower than the other strains that we could successfully control. The same data we could get from Denmark, where they did have a problem with community-associated MRSA, although not being the USA 300 strain. This was the definition that we used here, and we actually got the same information and the same data from 4 Danish hospitals in the Copenhagen area, and we did more or less the same analysis and came that here the community-associated MRSA had an RA value which was extremely low because there was hardly any in-hospital transmission. And again, the hospital-associated MRSA had more or less the same RA value that we found and that, for example, Ben Cooper has found in his studies. So we have now some information about, let's say, the reproduction numbers of MRSA in the hospital or in the community, not really in the community that we have that good calculations, but for hospital-associated MRSA, based on data in the Netherlands, Denmark, and UK hospitals, I didn't present this data, these are from Ben Cooper, it's always around 0.4 to 9.3, but below 1, and it's assumed that it's because of the repetitive admission of these patients that you get really successful transmission in the hospital. You don't need an RA value above 1 to reach that. For community-associated MRSA, we have estimates from Chicago. It's above 1. At least that's what we expect, but we can't really say what it is exactly because we have too many unknown variables to make that specific. In Denmark, community-associated MRSA in the hospitals is very low, and in the Netherlands, the animal-associated MRSA in hospitals is also very low. So to conclude on the MRSA part, there are different genotypes of MRSA, and they differ also in their epidemiology. There is genotype-specific transmission capacity for MRSA. Accurate quantification of the transmission capacity of USA 300 was, and I think is, still not possible because of multiple unknowns in the epidemiology. Readmissions are critical in maintaining endemicity of these hospital-associated MRSA strains. And in Dutch hospitals, the spreading capacity of this animal-associated strain is much lower, and in Danish hospitals, the spreading capacity of their community-associated MRSA strain is much lower than the hospital-associated MRSA. And there are some hypothetical valence factors in USA 300 and other community-associated MRSA strains that have been discussed and debated, but to the best of my knowledge, we cannot still not pinpoint to that factor or that trait being responsible for the success or the lack of success of some of these genotypes. Thank you very much.